All right, I should just have these memorized by this point. I've said them enough times. Sinew of sinew, when you sing a new tune of when you grew anew. Muscle, muscle tissue. All right, so here is our old friend, our Persian pal, Avicenna, from Iran, what is today Iran. And I dubbed him the Galen of the Islamic world, dating back to the 11th century, around the year 1020 or so. In one of his great tomes, he wrote, quote, "The power of locomotion is that which contracts and relaxes the muscles whereby the members and joints are moved, extended or flexed. This power reaches the limbs by way of the nerves, and there are as many forms of power as there are of movement. Each muscle has its own peculiar purpose, and it obeys the decree of composite sense." All right. So already a millennium ago, this gentleman had, in fact, almost exactly a millennium ago now, this gentleman had a clear sense of the afferent-efferent loop, the relationship between the nervous system and the muscular system, and how muscles work in tandem, as in groups, in coordinated fashion, obeying the decree of composite sense. Europe takes the next step forward 600 years later, 500 years later, with Vesalius, who I've already pointed out in the opening lecture. A guy from Flanders who was a physician, a doctor at the time. Physician not being what we think of it as today. I'm sure this guy was very adept at the use of leeches and other such devices of the time, trying to bleed off the ill humors that one disease or another may be caused by. But one thing that is undisputed is his contribution to our understanding of anatomy through his lovely and certainly exquisitely rendered anatomical depictions. All right. So the beginning of last lecture, I started with an image of a light microscope image of some muscle fibers with the motor neurons attached to them. I'm going to start today's lecture with the same image, but instead of a light microscope, this is an electron micrograph, a much higher resolution image. And we see here the neuromuscular junction. This synaptic baton, buton, B-U-T-O-N, or knob, buton being the French term that was more common in the 20th century. But the neuromuscular junction attached to the sarcolemma of the muscle fiber. And we talked about sarcolemma in lab yesterday. So just again, reviewing a little bit of the background that I've already covered. There are three types of muscle tissue. There's skeletal muscle, cardiac muscle, and smooth muscle. The skeletal system is only made up of skeletal muscle. Skeletal muscle is striated. Cardiac muscle is striated. Skeletal muscle is polynucleate, whereas cardiac and smooth muscle are mononucleate. Both skeletal and skeletal muscle is the only voluntary muscle fiber, whereas both cardiac and smooth muscle are involuntary, meaning the contraction of these muscle fibers is not under your conscious control. not explicitly. We can, through the cardioregulatory system and vagal output, influence the rate of depolarization in our heart. We can influence our heart rate, but we can't just cause our heartbeat to spontaneously start or stop. All right. So, muscle fiber, and I'm about to set up a paradigm that you're going to recognize at different points in the semester. And this is this paradigm of the functional element that we're discussing in the organ system that we're discussing, and its relationship to various layers of connective tissue. So we're going to, we'll see this in the central and peripheral nervous system that we discuss on Tuesday of next week. But for today, it's muscle tissue. So obviously, the functional component that I'm referring to here is the muscle fibers. And there are, in a muscle, in a whole muscle, there are 3 layers of connective tissue that follow a hierarchy. First of all, there is the epimysium. Epi meaning on the surface of, or external to. This is a layer of collagen that is external to the muscle fibers, the muscle as a whole. It acts as a sheath surrounding the muscle that helps separate the muscle from surrounding tissues, and in some ways acts as a way for that muscle to slide along the boundary that it has with surrounding tissues, be that other muscles, bone, or other tissues of whatever sort. So this epimysium kind of acts a little bit like the serous membranes that we discussed around the heart and the lungs, etc. However, they are not membranes in that way, and they're not making any kind of exudate. They're just this connective tissue, strong connective tissue sheath made out of collagen. It is loosely connected to the deep fascia. Deep fascia is the connective tissue sheath that surrounds our body beneath our hypodermis. All right. The next layer of connective tissue is called the paramecium. So you'll notice that we have a single muscle, but that muscle itself is bundled into these things called fascicles. Fascicle comes from the Latin word fasces. Does anybody know what the word fasces is? It actually relates to American governance. What's that? Yeah, okay, so you're right. That is, so very good. Fasces is the term that I use to describe face. In lecture, it's the word used for face. But that's the F-A-C-I-E-S. The fasces I am talking about is the F-A-S-C-E-S, right? Fasces, I believe that's how it's spelled. So if you look, I went to University of Virginia for my master's degree, and that is the land of Thomas Jefferson. Everybody bleeds red, white, and blue down there. So there are statues all over the place of the various forefathers of our nation. And along with these statues, you'll see there's one of George Washington on the campus on one of the central quads there. George Washington standing with a bundle of sticks, a bundle of little spears, I think, or arrows or something. I don't know what it is, but it's basically a bundle of sticks that has a ribbon tied around it. This is a Roman symbol of power related to the consul or the proconsul, which is were leaders in the Senate, the Roman Senate. It was a symbol of power that, for example, a person in a territory Roman territory would have when they are up on the deck holding court or whatever. But it's the same word origin here. So fascicles, fascicle, is simply a bundle of nerve fibers. So a whole muscle is separated into these fascicles, which are themselves bundles. It is the paramecium that helps to define the boundaries of the fascicles. All right. Epimecium defines the boundary of whole muscles. Paramecium defines the boundary of fascicles. And then we're going to see that the pattern continues. The paramecium ends up being the conduit for the neurovascular supply in a muscle. So you're going to have arterioles and venules passing through the paramecium, as well as the small nerve roots, the nervelets that are going to connect with the muscles and form neuromuscular junctions. Okay, finally is the endomecium. So each fascicle itself is simply a collection of muscle fibers. Muscle fibers are surrounded by endomecium. Endomecium is the most intimate layer of connective tissue, and it defines the individual muscle fibers. You are going to have some small capillaries now. We've gone from arterioles and venules to capillaries that run through the endomecium. This is where we find the myosatellite cells taking residence. Myosatellite cells are cells that are going to help repair damaged muscle cells. They are the cells that can fuse together. They are the cells that provide the multinuclei for the multinucleate muscle cells. These muscle satellite cells, these peripheral muscle cells, are there for repair of damage to the muscle fibers when they incur such. All of these muscle layers of muscle connective tissue, when at the terminus of a muscle, either the insertion or the origin, the muscle fibers themselves run out, and all that connective tissue continues though and comes together to form the common tendinous either origin or insertion, the attachment for that muscle. All right, so that's what a tendon is. It's just the continuation of all these connective tissue layers, the paramecium, the epimecium, paramecium, and endomecium. Mecium referring to muscle. Okay. If I get to it, I may or may not, but at the end of the lecture, I'm going to talk a little bit about amyolateral sclerosis, or Lou Gehrig's disease, a disease that claimed a number of people in my life, including my father. But if you look at a tissue section from a muscle that has been wasted by this disease, they almost look like tendons because the muscle fibers have withered away and died, leaving naught but the connective tissue behind. All right. So again, to be redundant, help you remember all this, skeletal muscle fibers themselves are exceptionally large, exceptionally long and large. Mesodermal, I cut out a couple slides about embryology that maybe were still in the PowerPoints that I had posted on Moodle, but I decided not to talk about. The ectoderm, mesoderm, and endoderm slide. I didn't talk about any of that. But mesoderm is one of the embryologic tissue layers that arises, that for those of you who go on will learn about elsewhere. But these mesodermal cells give rise to myoblasts, which are either going to differentiate into mature muscle fibers, or into these myosatellite cells that are there for repair. purposes. So when there's the fusion of all these mesodermal cells, we get these massive muscle fibers. They can be, some of them can be exceptionally long. You can have ones that are very, very long that go throughout the length of some of the longer muscles. And they can have hundreds of nuclei. So now we've established that they're voluntary, that they're multinucleate, that they're striated. I've talked about the anatomy of the striation. We're going to talk about the functionality of it in just a bit here. That's going to be the first half of lecture. The number I have here is these muscle fibers can be as long as 30 centimeters. So that's about a foot. A cell that's a foot long. Think about that. That's pretty remarkable in your own bodies. And in exceptionally tall people, who knows, maybe it's longer. All right, so just to recap that anatomy that we went through yesterday. The plasma membrane is known as the sarcolemma. And the action of a muscle fiber is going to start here. It's going to start by a depolarization in the transmembrane potential at the sarcolemma. This is what's going to start the contraction. It's going to spread across the sarcolemma and through these T tubules, these transverse tubules, T tubules. And the function of this is to spread this depolarization event throughout the entirety of the cell, reasonably simultaneously. All right. There are some really nice animations in the Moodle showing this. That if you were to watch those, it would probably save you the 5 minutes spent looking at those would probably save you an hour of listening to me talk about this. They show it visually quite well. So the cytoplasm of a muscle cell is called the sarcoplasm. In general, sarco is just I said it yesterday, it means meat, and it refers, it's the prefix that refers to muscle fiber. So the sarcoplasm is full of glycogen, and we'll see. Maybe I used to have a picture. It seems like I would have shown it by now. I must have cut it out. But I might have a picture that we'll see some of the glycogen. It's stained for glycogen, so you can see really how much glycogen is in the cytoplasm. For the benefit of all who might not know, could somebody tell me what glycogen is? Does anyone know what glycogen is? None of you? Okay. It's stored sugar, yeah. Glycogen is essentially starch, but it's animal starch. Starch is from vegetable sources. Glycogen is from animal sources. It's really just a polymer of glucose, beta-1,4 linked glucose with branches beta-1,6 linked. So it's just a storage of glucose. The transverse tubules, oh shoot, I didn't talk about it in lab yesterday. It doesn't matter, I'll talk about it now. So these T tubules in this diagram, they're painted this dun color, and I think they were blue in the model that I used yesterday, whereas the sarcoplasmic reticulum, the colors were reversed, but that's meaningless. So the sarcoplasmic reticulum is the functional equivalent of the endoplasmic reticulum in a typical cell. And it is the site for calcium sequestration. That means this is where we stash all the calcium. How do you suppose the cell does that? How do you suppose that happens? So we've talked yesterday about how the cell stashes potassium inside the cell and chucks all the sodium outside the cell. How did that happen? Yeah, active transport. We had pumps, didn't we? We had pumps that were burning ATP. So by extension, if the sarcoplasmic reticulum is where we're stashing lots of calcium, you would be safe in inferring that there are going to be active transport calcium pumps that are constitutively stashing the calcium away. All right. This endoplasmic reticulum acts as a sheath, bundling together a set of myofibrils. A myofiber is a single muscle cell. A group of muscle cells is a fascicle. All right. But within a myofiber are these myofibrils. And here's a myofibril. It is this collection of the myofilaments. So some of you may guess where I might be going with this in a moment. But myofibril has its own sheath around it that helps to define it. That sheath is made of endoplasmic reticulum with a little T-tubule lasso around it. So within the functional unit of a sarcomere, we have one segment of endoplasmic reticulum bounded on either end by T-tubules. So within the area of a T-tubule, the distal portion of this endoplasmic reticulum has a special name. We call it the terminal cisterna. Is that name on there? I don't know if you see it. But this is called the terminal cisterna. Cistern, like like on an island in a tropical place, would be a place for the collection of rainwater. A cistern. Terminal cisterna. It's on the terminus of the this portion of the sarcoplasmic reticulum. So two adjacent terminal cisterna with the intermediate T-tubule forms what is called a triad. All right, a triad is essentially an Oreo in terms of having or a sandwich. It's a T-tubule sandwich on terminal cisterna bread. That's a triad. Okay. Is that anatomy in there? The anatomy is important because it's going to structure function and all that. I'm going to talk about how this works. All right. So again, let's just continue to enforce the anatomy here. Here is a sarcomere from Z line to Z line. I don't remember who it was. Helped me remember the actual definition of Zusamin. But it means in between. So this is the boundary from one sarcomere to the next. So between two Z lines is a sarcomere. In the middle is this A band, A standing for anisotropic, meaning it does not rotate polarized light. And then the I band, the light band, is isotropic, meaning it does rotate optically. It's optically active. It rotates polarized light. Within the A band, there is this H band. This is a little bit lighter portion of the A band. This is where we only have thick filaments. All right. The I band, we only have thin filaments. It is this zone of overlap. Thank God, finally something has a name that makes sense. It is where the thin and the thick filaments overlap with one another. And this is where the action is. This is where the money is. I taught yoga last night, and 3 members of the baseball team showed up. Real nice guys, super muscle-bound, had a hard time touching their toes. And they were in the midst of strength training for their season. And I was telling them, and I'll show an image of it later, hopefully if I can get to it, of the power output of a muscle as a function of resting length, as a function really of zone of overlap. So optimizing the length of this zone of overlap for anybody who is trying to get more performance out of their muscle. You get more power if you're able to, out of a muscle without adding any muscle mass, just by getting the muscle to the right resting, by optimizing the zone of overlap. If this is too small, there's not enough rubber on the road, the muscle's been stretched out too much. If the muscle is over contracted, meaning like a person who's kind of muscle bound and not especially flexible, then there's not going to be there's not going to be anywhere for the muscle to contract to. All right, so you're going to be limited in the amount of power output that you can get from that muscle. Does that make sense? We'll see it in a bit. It'll make more sense. All right, so here's a cartoon that may be a little more clear. In this cartoon, the pink lines are the thin filaments, the purple are the thick filaments. Here's the M line, the middle, where everything is attached to. The Z line is the zigzag line that the thin filaments are attached to. There is this wavy green line is titin. And titin is a protein that helps keep it all lined up and keeps it from being a bowl of spaghetti. All right. It helps with the return to normal resting length as well, but mostly it just keeps the myosin on track with the actin. Here are some cross sections that may or may not be useful to you. There's no unique point to be made here, except that if you were to look in cross section, so where's the zone of overlap? You have here the purple is the myosin. These are the motor proteins, and the red are the actin. These are kind of the train tracks, the tracks that the motor protein is going to work on. And we can see here that it's in this hexagonal close pack array. It's optimizing spatial density here. So you have one motor strand, one myosin strand surrounded by 6 actin filaments that it can grab onto and move forward. All right, so let's keep getting closer. Closer look. What are these thin filaments? Well, they're made of F-actin, F standing for filamentous. This is a spiral protein. Spiral protein is basically beads on a chain made of G-actin, G standing for globular. It's in the globulin superfamily of proteins. So it is the active site in this picture depicted as a yellow spot in this cartoon. This active site, which is what comes into contact and is sticky to the motor protein in myosin. That's where the myosin is going to grab onto and move it. There's a couple other important proteins. First is called nebulin. Nebulin is this thin cord that runs down the middle that the G-actin helps. It helps the G-actin adhere, and it forms the F-actin strand. And then, importantly, these two proteins, tropomyosin and troponin. First of all, tropomyosin is this double-stranded protein that lays right on top of the G-actin active sites, and in fact occludes them, prevents myosin from interacting with them. Its placement there on the G-actin site is regulated by another protein called troponin. So troponin holds tropomyosin in place on the G-actin, on the F-actin, on the active site. And it is responsive to the presence of calcium, all right? So if calcium is there, troponin will bind calcium, and it's going to change the orientation of tropomyosin with respect to the F-actin. So you can already see here that we have all of the elements in place. We have an excitable membrane that penetrates the cell, that goes through these T tubules that are right adjacent to the sarcoplasmic reticulum, which is full of calcium, right? And next to muscle fibers, where there's a protein called troponin that wants to bind to calcium, who regulates the interaction of myosin and actin. We're setting the dominoes up. There's a couple pieces, but we're almost there. A couple pieces to go yet, but we're almost there. Thick filaments. So, before I go there, I'm just going to point out one thing to help you remember the I-band. So I said it rotates, it's isotropic. It rotates polarized light, right? Well, look at this. That makes sense, right? It makes sense. There's a handedness to the twist of this, to the twist of the F-actin. It's going to, as light comes, it's going to rotate light in a particular handedness because the spiral is all going in the same direction. This is not the case for myosin. Now, they because this is an artist's rendition, it looks a little bit like a spiral here, but myosin is not isotropic. It's anisotropic. So there are these myosin subunits that there's the thick filaments of myosin that are made up of these myosin subunits. The subunit itself, so don't be confused why it says twisted myosin subunit. This is a myosin subunit. Here's the myosin molecule. One of these combines with many others to form this anisotropic bundle. The molecule itself, a single myosin, has this twisted tail, and then there's this hinge region here, and the myosin head. It's the myosin head group that's going to interact with the actin, and it's this hinge that's going to act as a spring and help it to crawl along the actin subunit. All right, and there's the depiction of Titan, as I had suggested, that helps the recoil after stretching. So that's pretty much all the players we actually need to get the basics of it. I'm going to just highlight the fact that there are a lot of other players in this story. There's at least 7, probably many others, that are responsible for connecting the contraction of these myofibrils with the outside world, with what has to be the connective tissue, right? Because unless you're transferring that force to the connective tissue sheath to pull on the tendon, you're not going to generate any force. So you have to connect the contraction in these filaments to the connective tissue on the outside. And there's all these different proteins, these linker proteins that make that happen. One of them is called dystrophin because abnormalities in this protein are responsible for the condition called muscular dystrophy that maybe some of you have come into contact with in your life. And thus giving the protein its name. All right, so this is kind of, I'm not a physician. I'm a PhD with an emphasis on the doctor of philosophy. I am a bit of a philosopher, and I find these kinds of things interesting. Muscle tissue, you could perhaps appreciate this, that there is a scale invariant self-similarity or roughness to the system. That's what fractals are. And there is a fractal aspect to muscles. If you look at the analogies as you head down the superstructure, we have a skeletal muscle that's surrounded by epimysium, a muscle fascicle that's surrounded by paramysium. which is made of muscle fibers that are surrounded by endomysium, which is made up of myofibrils, which is surrounded by sarcoplasmic reticulum, which is made up of sarcomeres, which is itself made up of the various myofilaments. So there's this hierarchy that is a sort of repetitive motif down the size scale that I find to be pretty intriguing. Oh, yeah, here's the picture I was thinking of. So right here, these green vacuoles are droplets, vacuoles of glycogen in a single cell. These are not individual muscle fibers. They are individual myofibrils within a single muscle fiber. So this is some kind of a transmission electron micrograph, 88,000X, big. Yeah. So I guess the point here is that muscle fibers, for them to work, we need ATP. We're going to need some ATP because the energy that causes that spring to in the hinge region of the myosin to contract, that energy comes from ATP. So myosin is an ATPase. It's a protein that hydrolyzes ATP to ADP and inorganic phosphate, thus releasing that energy and causing the contraction. For there to be aerobic contraction of a muscle, we're going to need to use oxygen. All right? And whether it's aerobic or anaerobic, we're going to be breaking down glucose. It just depends on how efficient we're going to do that. All right. So, here's the sliding filament theory. The basic idea is that there's this zone of overlap, and when a muscle starts to contract, the myosin runs itself over the tract of the actin and pulls the muscle fibers together. Can I have 4 volunteers who want to get out of their chairs? Just come on up. 4 people who want to stand up and don't mind holding hands. We can do some hand holding. All right, I'm going to be on the end of this. So everybody spread yourselves out from one end to the other and extend your arms like me so that we're touching. So here, let me think about this. You and you, Casey and Alan, you guys are actin. And the rest of us are all myosin, okay? And so they're just going to hold their arms still, all right? They're not going to move at all. And as a myosin, your job is to go like this. Just grab their arm and pull yourself along their arm. And we'll see what happens. Everybody obviously knows what the outcome of this is going to be, but let's contract. Wait, grab hands there. So calcium is in the thing. Let's contract. Come on, myosin. Do it. Well, this end of the muscle is contracting. Okay. So your job is to grab his arm, bend your wrist, and pull him towards you. You want to pull him, and her too. Pull her towards you. And you do the same, right? You're going to try to work your way along that person's arm, pulling them towards you. All right? All right, let's try it again. Contract. So there we go. You can see more or less, okay, right, and it's pulling me down this way, isn't it? Yeah, it's me and Annabelle are getting pulled towards the middle, and the chain is shortening. These two didn't do anything, they were just passive tracks that the 3 myosin molecules were running along as we were contracting. Thank you very much. Good job. So if you look at this, you can kind of see how this works. Contraction, relaxation, contraction, relaxation, relaxation, contraction, relaxation, contraction, relaxation. You want to see a contraction again? Contraction, relaxation. Yeah.
So are neurons what stimulate?
Yeah, we're going to get there. We're going to see that we haven't even knocked the dominoes over yet. Yeah, we haven't knocked them over, but we will. In fact, perfect question. You've set me up just perfectly for this slide. All right, so here is an overview of what actually happens. There's going to be neural control. And we're going to talk about exactly how neurons work in the CNS chapter. But a stimulus comes down the chain. There's going to be a synapse, a release of acetylcholine. And there's two phases. There's excitation. There's actually three phases: excitation, contraction, and relaxation. And there's this excitation-contraction coupling that needs to happen. So there's two events. There's the excitation, which is an event that happens at the motor end plate, where the synaptic bouton in blue up there with the acetylcholine vesicles releases the acetylcholine. It causes a depolarization in the sarcolemma, which travels across the surface. This is the excitation. It travels across the surface of the sarcolemma to the T-tubules. This is going to cause a release of calcium in from the sarcoplasmic reticulum. The calcium is going to bind to troponin, which is going to roll tropomyosin off of the F-actin. And then we're going to go through that business that we just did up here, the contraction of the muscle fibers. And this is going to generate tension. All right, so that's an overview, and I'm going to go through it step by step by step, so don't worry that I flash through it. Am I going too fast? No? Okay. Excitable membranes. Who doesn't love them? Okay. 4 major phases. Okay, there's 4 major phases of contraction and relaxation. So there's the excitation-contraction coupling, the contraction, and then the relaxation. And I'm going to walk through each of them one at a time. I just in the previous slide, I just described each of those, so I'm going to go through them in detail here. First, excitation. Well, let's look at the scene. We have this neuron at the motor end plate. It's ready to go. It's loaded with all these synaptic vesicles full of acetylcholine. Acetylcholine is a neurotransmitter. Here's the synaptic cleft. On the postsynaptic membrane, here's the presynaptic membrane. Postsynaptic membrane, we have it loaded with all these acetylcholine receptors. These are chemically gated. Sodium channels, and they're embedded in an excitable membrane. Why is it excitable? Is it excitable because it has acetylcholine receptors in it? No. What makes it excitable? It's excitable because it has voltage-gated receptors in it as well. All right, it has voltage-gated receptors as well. The acetylcholine receptors are going to cause a local graded potential that's going to immediately trigger an action potential, because this membrane is also excitable membrane. It's got voltage-gated sodium channels as well. So there are chemically gated acetylcholine receptors here, and then there are voltage-gated receptors that are going to start the action potential. All right, you get that? Because the sensitivity of this synapse can be modulated based upon the number of acetylcholine receptors. You can downregulate acetylcholine receptors and make the synapse less sensitive. You can attune that. Any other picture here? No, I think that's everything that we need in that picture. So let's actually do it. Step 1. We've got an action potential that arrives. It's coming down the axon from the neuron. The acetylcholine's ready to go. And the depolarization of this membrane causes a membrane fusion event that involves calcium as well. Remember I told you that back when we first started talking about calcium on day 1? Or maybe it was that day 2? No, it was day 1. But that's beside the point. We release acetylcholine into the breach here, into the cleft, synaptic cleft, and it's going to bind to these chemically gated sodium channels. When that happens, we're going to depolarize this membrane locally and set off the electrically gated channels. When those electrically gated channels get tripped, then there's an action potential. All right? So here's that acetylcholine receptor. It gets hit with some acetylcholine, it opens up, and we get the flow of ions. Now, here are the voltage regulated receptors. Channels, I guess, is probably the best word. It's not a receptor. We have the chemically gated receptors, and then the voltage gated channels that make the membrane permeable to these ions. So sodium at first is going to be, just like I talked about yesterday, sodium is going to be the first one to open up. It's going to depolarize, and then it's going to close, and the potassium is going to open up, and we're going to repolarize the membrane, just like we talked about yesterday. All right, so everything I talked about sodium and potassium holds true today in the muscle. All right, so then this action potential has been started, and it begins to propagate away from the motor end plate. And meanwhile, acetylcholine diffuses off of these receptors and binds to another enzyme that's just constitutively present in the cleft called acetylcholine esterase. Acetylcholine esterase cleaves the ester bond between an acetyl group and choline, which is a small organic molecule. And these fragments then get recycled back into the presynaptic bouton. Everybody cool with that? So that's excitation. That's excitation. Now we want to couple that with contraction. We're not at contraction yet. This is excitation-contraction coupling. So its action potential travels down the T-tubules. Here we are in a triad. Here is the T-tubule, and with the adjacent terminal cisterna on either side of it. This depolarization in the T-tubules causes a similar depolarization in the sarcoplasmic reticulum, which opens up calcium channels and allows calcium to flood out of the sarcoplasmic reticulum into the sarcoplasm, where the myofibrils are. Myofilaments, pardon me. Or myofibrils, the same, I guess. All right, so that's excitation-contraction coupling. So the calcium comes out into the sarcoplasm, and then it's going to bind to troponin. It's going to bind, calcium binds to troponin. Troponin rolls tropomyosin off the active site. All right? When this happens, we are now able to form a cross bridge between myosin and actin. And we're going to start talking about contraction. Each step in contraction sets up the ideal circumstance for the next step. All right? So they lead one into the other consequentially. Step one. Do I yell at you guys? Am I speaking loud enough, too loud? Am I the right? Sometimes I feel like I'm yelling at you. No? Okay. I don't want to mumble. the resting sarcomere. Here it is. There's no calcium in place. The point of this slide is to show you that here's this myosin head group. It's cocked back. It cannot bind the active site on G-actin yet because troponin has not bound calcium, and thus tropomyosin is still covering the active site. Looking at the head group, what do I see there? I see an ADP molecule bound to it along with inorganic phosphate. There's essentially an ATP that's already been hydrolyzed. Nothing's happened yet. The resting state of the myosin has it cocked back with an ATP already invested. There's a sunk cost here. We've already sunk the cost of one ATP before we've even asked the muscle to do a single contraction cycle. What might be the advantage of that? Yeah, rapid start. Thank you. Exactly. All right. So let's bring in some calcium. Calcium comes in, binds troponin, which rolls tropomyosin off the active site of the F-actin. Now we're able to have cross-bridge formation. Now I'm going to, do I want to do this? Do you guys want a little bit more nuanced biophysical picture of it? This is the world I live in. I love it. I'm going to do it. Why not? So this is a cartoon. This is a cartoon. But in the real world, there is all kinds of cytosol there, right? Sarcoplasm. That's an aqueous environment. Now, in a glass of water, I'm not going to take more than 2 minutes. I'm going to cut myself off after 2 minutes. In a glass of water, there is the bulk water solvent structure, right? And water, we all know, water hydrogen bonds with itself and forms these transient chains of hydrogen bonds which break and reform in different ways. And then you put a solute in the water. You put a solute in the water, and the water has to organize itself in some way around that solute. And so solutes that have the least organizational burden, or the word that I would really like to use is entropic burden, right? The least entropic burden in terms of how it's going to limit the degrees of freedom that the water molecules have, is going to be easier to get it in solution. All right? So things that dissolve really easy are ones that allow the greatest degree of freedom for the water in the solvation sphere right around that solvent. There's a sphere of solvent molecules around that solvent, and it spreads out into the bulk solvent. Because if you have a group of highly ordered water molecules right around the solvent, then there's going to be another shell around it that's a little bit less ordered, and so forth until you get back into bulk solvent, right? I'm going to set that aside for a moment. Consider how binding partners in a biological system may work. You have, for example, a T cell, perhaps only one T cell that recognizes some obscure pathogenic antigen in your body. And it lives in your toe. Maybe it's in your toe. You prick your finger, you get some kind of infection over here that only this antibody down there is going to recognize. Does every T cell in your body have to go up and see if it fits that key before we get an immune response? How do binding partners, things that are supposed to bind with one another in biological contexts, find each other at a distance? It's not like they're looking and they can see each other in a crowd. Right? It's not how it works, although kind of it does. So the point that I'm making about solvation spheres, as things evolve to have complementarity, structural complementarity in binding, it's not just like any old key and any old lock, another constraint on the structural uniqueness of those two surfaces is how each of those surfaces control the solvation sphere around it, and how easily the specific binding partner that it has is able to desolvate that surface and come and bind, versus other solutes that are not supposed to be binding, right? So evolutionarily, binding partners develop in such a way that they affect the bulk solvent so that it can easily desolvate and approach from a distance. That's what's happening here. That's what's happening here. As soon as this is revealed, as soon as this active site on G-actin is revealed, the specifics of the structure of that site enable this part of the myosin to really easily desolvate it and come close and attach as a binding partner. All right? In fact, it increases the degree of freedom of the solvent molecules that are here solvating these two structures in the first place. You guys capiche? A little bit? You don't have to. It's a nuance for the people who want to think on a little bit higher order. It's okay. All right, let's keep going. So cross bridges get formed. And as soon as that cross bridge forms, the act of forming that cross bridge is going to cause a slight conformational shift in this protein, in the myosin head. That shift is going to do two things. It's going to kick off the ADP and the pi. When it kicks off the ADP and the pi, the inorganic phosphate, when this comes off, this is the stopper that's holding the spring back from contracting. When ADP comes off, the head pivots. The head pivots. This is where the actual contraction is. This is the step that actually moves the two strands with the relationship to one another. When the head moves forward at the hinge region, we have another conformational shift, which now makes the actin head group, the myosin head group, not want to bind actin. It's not in a favorable position to bind actin any longer, so it disassociates. It detaches. When it detaches, an uncocked, unbound head of a myosin, it really wants to bind ATP. It's in the perfect position for ATP to bind. When ATP binds, the act of binding causes ATP to be hydrolyzed. As ATP is hydrolyzed, that energy causes a conformational shift again in myosin. The energy gets stored in the hinge region once again. And we have opened it back up and ready to start the cycle again. So now, if calcium is still in the scene, if calcium is still here, and the active site on G-actin is still available, we're going to go through this cycle again. And we're going to continue to go through contraction cycles multiple times until the calcium is not there any longer. Does that make sense? It's pretty straightforward, I think. There's just a lot of steps in it. All right. Yeah, so I got this. Yeah, I get these like silly slides. So is it contracting from one end to the other or both ends? I don't know, I guess which way you think up is. But you can see what's happening here. So in the top, in this picture, we see that there is a large I band, smaller zone of overlap, and a larger H band. Now we go down here, the I band and the H band are really small and the zones of overlap are really large, right? Because the two fibers have moved over top of one another and the muscle fiber has shortened. Is anybody mystified at all? Is anybody bored? Am I going too slow? All right. So how long does this last? Depends on a number of factors. What's the duration of the neural stimulus? How much free calcium is in the sarcoplasm? Are we calcium limited? All right. If you're calcium limited, then it's going to be a shorter contraction. And then importantly, I'm going to spend probably most of the rest of the class talking about the availability of ATP. ATP is important. You got to have ATP for a lot of parts in this chain. Let's think about that. You got to have ATP. So let's go back all the way back to the motor end plate. What kind of proteins do we know must be in that membrane there? Let's list them. There's more than you may realize initially. So we've got, just call them out. What are some of the proteins we remember or infer must be there? What are the ones I told you about explicitly? I said, what's the neurotransmitter? Acetylcholine. So we know there's going to be acetylcholine receptors. Acetylcholine receptors. All right. What else is there? There's going to be voltage-gated sodium channels. There's going to be voltage-gated potassium channels. What else? From yesterday, if we know that there's voltage-gated sodium and potassium channels, what other proteins are going to have to be up there? Say it, yeah. Sodium, potassium, ATPase. The pump. It's going to have to be there, right? And there's probably going to be some sodium and some potassium leak channels. Because this is on constitutively, as long as there's ATP, right? That's just on. So to keep it at the right level, we're going to have leak channels in the right balance with the number of sodium, potassium, ATPases. All right. Well, this baby needs ATP, right? That thing needs some ATP. And we already established that calcium in the sarcoplasmic reticulum is going up a gradient. So that's going to need some ATP too, huh? And we know that myosin is an ATPase, so that's going to need some ATP. This whole scene is dependent upon ATP at multiple points in the chain. Look at that. Look at how many different proteins have got to be up there at the end. And there's more than that, too. All right. Well, let's talk about relaxation first. Who doesn't like talking about relaxation? So when the nerve stimulation cuts out, the acetylcholine diffuses off the receptor, gets hit by the acetylcholine esterase, choline gets recycled, and the membrane starts to reset itself. When that happens, the calcium channels in the sarcoplasmic reticulum close, and the constitutive calcium pump takes over. It's called the rhyatidine receptor, I think is the name of it. So what's interesting is for this to happen, for muscle relaxation to happen, we need ATP. We need ATP for muscle relaxation to happen. If we don't have ATP, we can't get the calcium out of there. You can think about what consequence that might have in an athlete who's extremely ATP depleted. All right? You can think about what that might mean for an athlete who's extremely ATP depleted. and what might happen in their muscles. Also, we need ATP to recock these heads for the beginning of the next contraction cycle, to reset the myosin. All right, so not just ATP. There's a bunch of other things. There's elastic forces. So we talked about titin, the elastic elements of tendons and ligaments, etc. It helps bring a sarcomere back to its resting level. There are opposing muscle contractions. So he's like, you've like contracted these muscles. Oh, I can't. Ugh. Use your tricep. Tricep can help. Opposing muscle groups help lengthen a contracted muscle. And then when you can't fight anymore, there's always gravity. Just give up and let gravity do it for you. All right. So here's sort of an overview, a review of the whole thing, what I've been talking about for the last hour. We have, I'm going to go through the whole thing from start to finish. I'm just going to say it out there. So what do we have? Unless somebody else wants to try. Does anyone want to try? Does anyone think it's in their brain yet? Why don't I do it for you? We have a neuron that sends an action potential to the motor end plate that releases acetylcholine into the synaptic cleft. Acetylcholine binds the acetylcholine receptors, causes a localized depolarization at the motor end plate, which trips in the voltage-gated sodium channels to open and initiates an action potential, which spreads across the sarcolemma into the T-tubules, perforating the body of the muscle fiber, causing the concomitant depolarization of the membrane in the sarcoplasmic reticulum and the resulting release of calcium into the sarcoplasm, which is going to then bind to troponin, causing tropomyosin to roll off the active site of the G-actin and allowing the myosin head groups to form a cross bridge and contract and go through the contraction cycle, where there's the release of ADP, the hinge region contracting, and then the breaking of the cross bridge, and the rebinding of ATP, and going through that cycle until we hit resting conditions, which is characterized by the diffusion of acetylcholine away from the acetylcholine receptor and its recycling by acetylcholine esterase, and then the release of the excitation pressure on the sarcoplasmic reticulum and resequestration of calcium into the sarcoplasmic reticulum, with then the diffusion of calcium away from troponin and the replacement of tropomyosin onto the G-actin active site. Whew. That's it. That's what a muscle contraction is. It seems so easy, doesn't it, when you do it? But those are the steps. It's a pretty amazing system. Is that a new story? Is that an old story to anybody? Is anyone, the few of you who've had A&P before? You guys are probably looking for more from me. All right, rigor mortis. Yeah. So rigor mortis is a fixed muscular contraction that happens after death. And this happens because we're out of ATP. Why? Because you're dead. The ion pumps don't work anymore. We can't put calcium back in the box. And troponin continues to be bound to calcium. Tropomyosin doesn't do what it's supposed to do. And you get this cross bridge formation, but there's no ATP around to help kick the cocked head off the myosin. So you're in this like rigor mortis, the stiffness of death. So you have a big calcium buildup in the sarcoplasm. All right, that was my death metal slide for the class. All right, so in a single muscle fiber, there is, because it's dependent upon an action potential, the all or none principle is in play here. It's in force in a single muscle fiber, not in a muscle, not in a fascicle, but within a single muscle fiber. If it gets stimulated, that muscle is given its all. That muscle fiber gives its all. Just that one cell. All right. So as a whole, a single muscle fiber is either contracted or relaxed, not sort of like halfway there. All right. The tension that a muscle fiber can build is dependent upon that a single muscle fiber can build is dependent upon the number of pivoting cross bridges that muscle fiber has. So not all muscle fibers are built the same, right? Some are more efficient at doing their job than others. And we're going to talk about a couple ways that's true in a moment. Importantly is the overlap between the thin and thick filaments. This is what I was talking about to those baseball players yesterday. So in other words, what's the resting length at the time of stimulation? Is that muscle fiber in the optimal length to give the greatest force output for energy expenditure that it's using? All right. So amount of ATP in is not linearly correlated to tension production in the muscle fiber. There's factors that are going to increase or decrease the efficiency of the energy that you're burning in terms of ATP and the tension that a muscle fiber is actually producing. And then last is the frequency of stimulation. So remember, I was talking about, I was probably confusing some of you, torturing others yesterday when I was talking about amplitude modulation versus frequency modulation, right? So the informational content here is in the frequency of stimulus from the neuron. You know, the neuron can't tell you like, get moving or get moving. It doesn't yell or whisper. It just says, get moving faster or slower, more times or less times. The frequency with which there are subsequent action potentials. All right. So here's the slide that I've talked about a couple times already. That I think this is a pretty important concept for any person who is interested in getting the most out of their muscle performance. So what we have here on this is length of a muscle fiber, all right? Length of a muscle fiber. And really, actually what it is, it's length of the zone of overlap, which is a proxy for length of the muscle. So it's the length of the zone of overlap. And in the other dimension, in the y dimension, we have the tension that muscle fiber can produce. All right, how much tension, how much power can that muscle fiber put out? And so here, this would be the muscle-bound weightlifter that never does any stretching, never ever does any stretching. Okay, and that is a highly contracted muscle in the resting state. A highly contracted muscle in the resting state. And there's nowhere for that muscle to go at all. There's nowhere for it to go. There's lots of rubber on the road, but there's no road. All right? On this end, this is actually a torn muscle. Here's what this is. This muscle has been extended so far that there is no overlap. There are no myosin heads that are even able to form cross bridges. All right? So you're not going to be able to generate any force on this end either. Where you want to be, this is normal. This is normal. Most people are in here. Most athletes are in here. most people that spend all their time doing yoga and nothing else maybe are over here. I don't know who's over there. But this is where you want to be, obviously, if you're trying to get the most out of your muscles. You have enough overlap between the myosin head groups where you have optimal overlap of the myosin head groups, but there's enough of the stalk of the myosin left over that gives you plenty of room to have a strong contraction. All right? Any questions about that? Pretty straightforward. All right, so 3 phases of a twitch or a fiber contraction. So a fiber contraction is another word for it is a twitch. A twitch is a single, not a single contraction cycle, but a single contraction event, a neural contraction event. All right. So there's resting. Then there's some kind of stimulus. You're going to have this latent period. Why do we have a latent period? In terms of milliseconds, this is a couple milliseconds. So a millisecond is 1/1000th of a second. That's a pretty short period of time. 2 milliseconds is this latent period. What's that mean? Why is there a latent period? What is that? Before anything starts happening. Yeah, what do you think? Yeah, well, no, because stimulus, close. You're thinking the right direction. So stimulus is right here. Stimulus traveling down the axon is over here. Stimulus hits the motor end plate there. What do you think? Excitation and excitation-contraction coupling. Yeah, those 2 steps. Like you heard me, it took me like 3 minutes to say all that stuff. It takes your muscles 2 milliseconds to do it. But it does take a little bit of time to get the calcium in the sarcoplasm. All right? So then the calcium's there, boom, whoosh, contraction happens. We build muscle tension. All right, right around here, somewhere in here, acetylcholine is not being released any longer, and it's diffusing off of the acetylcholine receptor, being recycled, and we plateau and we begin relaxing. The tension begins to come down as calcium is resequestered and the muscle begins to return to a resting length. All right, so the 3 phases: rest, contraction, relaxation. All right, so the length of a single twitch varies dramatically. It varies dramatically, dependent upon a number of factors, primarily whether it's a fast twitch, a fast glycolytic, or a slow oxidative muscle. There's different types of muscle fibers, and we're only going to get into that a little bit because this is an introductory class. But so, for example, here's an eye muscle, which is a fast glycolytic muscle, real short time contraction. So around 7 milliseconds before it goes into relaxation. And then the soleus. Remember I talked about the gastrocnemius and the soleus yesterday. Those are the muscles of the calf. The soleus lasts, a single contraction can last up to 1/10 of a second, which is a pretty long time for a single muscle fiber when you're talking about molecular activities. Molecular activities happen on extremely fast timescales. All right. Why? I'm not expecting you to understand at the moment, at least, why, to be able to answer that question in terms of the specific molecular underpinnings of it. But just rationalize that from an evolutionary standpoint. Why would an eye muscle have a very rapid time course of contraction, whereas a soleus may have a much longer time frame for contraction. What do you think? What? Yeah. Yeah. When you blink, why do people blink? Not just to keep their eyes moistened, but we want it to be quick because we don't want to miss anything. And more importantly, we don't want to miss anything when like a werewolf is like coming in the door over there, and whoa, you got to like move quick, right? Yeah, you want to see that right away. Whereas a soleus, this is a muscle that is going to be a aerobic muscle, a slow oxidative muscle that's going to want to burn. It's going to be geared towards oxygen consumption and slow aerobic power output, right? So it's not going to be one that is rapid. And we're going to rationalize that in molecular terms in a bit. Yeah, so.
How does the twitch work?
Yeah, great, cramps. I have a question. I don't use it every year, but sometimes I use it. Talking about the origin of muscle cramps and the fallacy of lactic acid buildup. Really, what happens is, there's a couple different types of cramps, but the kind of cramp that you get when you just run a really long race or you swam a really long race and your muscle begins to cramp up is it has to do with the calcium receptors on the sarcoplasmic reticulum. They become leaky is what happens, and calcium is not able to be sequestered efficiently in the sarcoplasmic reticulum. So you're having sarcoplasmic calcium that should not be there, and the muscles are not able to relax properly. Is really the answer to that question. Yeah, that it doesn't necessarily have anything to do with the. So what did your question relate to the twitch again? I'm sorry.
Well, I was thinking that's.
Yeah. I would say a calf cramp is, a calf cramp happens because these are the large muscle groups that are repetitively called upon during some kind of athletic activity, right? I mean, your eye muscle. What are you doing with your eye muscle? Your eye muscle is not bearing any kind of load at all. The soleus is bearing a huge load, and so because it's bearing a huge load, the frequency with which you hang on. I'm going to get to some things that make it clear to you. So I don't want to re-explain myself here. All right. If you still don't understand, I can answer it later. Trapé. So a trapé is this stair-step increase in twitch tension. All right. So you have stimulus here, and it relaxes, and then another stimulus, and we get a little bit higher tension, etc. And we reach this maximum tension in trapé. So what the specific level of this tension is dependent upon the frequency of action potential that's coming in. All right, the frequency of the action potential is what's going to have the informational content of what the maximum tension is during a twitch. So this is due to repeated stimulation immediately after the relaxation phase. This is going to cause a series of contractions that step up to some sort of maximum tension level that is dictated by the specific frequency of stimulation. All right. So then we can, if we're going to like slam those stimulations, the frequency higher, if we're going to go to a higher frequency of action potential, we're going to get wave summation, where the tail of one overlaps, the leading tail overlaps with the trailing tail of the previous contraction. Does that make sense? So this leads to this case of wave summation, where you can step higher. You can step higher up the tension profile. You're going to get to this point where you get to incomplete tetanus. So tetanus means you're not allowing the muscle to ever get back to baseline relaxation. You're not allowing it to get back to baseline relaxation. But this is incomplete tetanus because the muscle physically can reach higher level of tension, but it's already in tetanus. It's never getting back to ground zero. All right? So we've reached a level of incomplete tetanus. What that level is dictated by the frequency of action potential stimulation. And we're going to push it even further. We're going to increase stimulation from the action potential, from the nerve, the motor neuron, even higher, to the point now where the leading tail of a subsequent contraction is bleeding back into the leading edge of the previous contraction. All right, so before we're even able to hit the relaxation phase of one contraction, we're going into a second cycle. This happens because the excitation phase is able to reset itself much quicker than the contraction phase. All right. So acetylcholine can be broken down and recycled, and the neuron, the upstream neuron, can get itself, through its refractory period more quickly than the muscle fiber can reset itself. This leads us to tetanus, what's called tetanus. Tetanus is a lack of contraction in a muscle. Muscle is not able, contraction, relaxation. Thank you. A lack of relaxation in the muscle. You get this maximum level of muscle fiber tension. It's not pleasant. All right. So let's step now up one level. Let's go up from the muscle fiber up to a group of muscle fibers, and talk about how we generate tension in a whole muscle. This is dependent upon the number of motor units that we deploy. So in this diagram, I've got three different motor neurons. All right, three different motor neurons, and they're all in the anterior horn of the gray matter in the spinal cord. There's the neurons. And each of those three neurons sends out their axons. One motor neuron is going to synapse on a group of muscle fibers within a single fascicle. So let's think of this as a fascicle. It's not a whole muscle. It's a fascicle. It's a group of muscle fibers. Within that fascicle, a certain number are all going to be under the control of a single motor neuron. That means when that motor neuron fires, all of those muscle fibers are going to fire. They're all going to hit all or none. And to generate tension in a muscle, our nervous system is pretty smart. It doesn't just fire off all of these all at once. We are able to, depending upon how much muscle tension we want to consciously generate, it can stagger. Those motor units. It can recruit a subset of them in successive waves. So within the perspective of the whole muscle, they overlap, the tension that each motor unit is generating overlaps so that the red line is the observed tension in the muscle itself, in the tendon of the muscle. Is this making sense? Yeah. So now if I'm sitting here and I'm going to pick up this rod, I don't need to recruit all of the muscle, the motor units in my bicep to do that, right? I'm just recruiting a small subset of it. Whereas if I were to try to lift this table or something, that is going to require me to recruit a vastly larger number of motor units at the same time, all right? And if you push a whole muscle to its limit, you are effectively recruiting the bulk of the motor units all at the same time, all right? So you're pushing this together this way to get an increase in tension. This makes sense? It should be pretty intuitive. All right, so let's see how we can pay for all this. Our bodies are really just big bonfires. What's that book by Tom Wolfe, Bonfire of the Vanities? That's it. The way our bodies work with energy, it's just combustion. We're taking carbon, a carbon source, completely oxidized carbon source, and we're oxidizing it, giving off carbon dioxide and water. We're taking unoxidized carbon, combining it with oxygen, and getting carbon dioxide and water out of it. That's what combustion is. Our bodies do it by eating carbon, inhaling, and then exhaling, inhaling oxygen, exhaling carbon dioxide, and then the water just goes into the soup of our bodies. All right. It is this process of burning carbon, primarily in the form of glucose, that enables us to regenerate ATP supplies. This is probably review for at least half of you, probably more than that. Okay. So I'm going to talk about a few things. I don't know if this is review or not for those who've taken anatomy before, but I'm going to start by talking about the phosphagen system. So we said that phosphate-rich adenosine is the currency of energy that we need, specifically ATP. But coming up with creative ways of storing phosphate energy, phosphogenic energy, is smart, and our cells are smart. So this is one of them. This is part of what's called the phosphagen system. There's a couple components of the phosphagen system, but here's one. So any muscle bodybuilder is probably seen creatine. They sell all these creatine supplements you can pay a lot of money for and shovel down. And the way this works is we can store phosphate, readily available phosphate, in the molecule of creatine as creatine phosphate. We can just store the phosphate there and get it out of the bank quickly. All right, that's all that this is. So this creatine phosphokinase enzyme takes ATP when we are flush with ATP. Say you're just chilling out, you're eating your granola bar, whatever you're eating. You're flush with ATP. We've got creatine in the cells. We're going to pack a bunch of this with phosphate. Now, is it long-distance runners? Is it cross-country skiers? Is it swimmers? Is it people like that are thinking about this? No. It is the aerobic athletes, the weightlifters, maybe the football players, the people who need short, powerful, rapid bursts of energy. all right, who use this, because this burns out quickly. You burn this out quickly. This is not energy for the long haul. So what I'm going to be talking about is this gradient of time scales that we're using to pay for energy contraction. And creatine phosphate is on the shortest end of that spectrum, all right? We can just quickly throw some phosphate into creatine and get it out rapidly, okay? Maybe I need just a powerful burst of energy for a brief period of time, and I don't want to burn a bunch of oxygen and other fuel to get it. All right. Here's another example. So maybe we need some ATP right away. Right away. This is like taking a second mortgage on your house. This is an emergency activity. Because what we're going to do is we burnt a bunch of ATP and there's a bunch of ADP around. But we can squeeze a little bit more energy out of that extra phosphate in ADP by taking an ADP and transferring one of its phosphates to another ADP, giving us an AMP and one ATP. All right, so we can take two ADP that have been burnt and get one more good ATP out of it. We're wringing the most out of our phosphate. And this is myokinase or adenylate kinase, ADK. It goes by different names. That does this. All right, next is aerobic metabolism. Again, I apologize. I'm sure this is probably redundant for many of you. Anyone who's had central metabolism before. This is like central dogma here. This is central dogma. All right. This is you guys chilling out. All right. You have had breakfast. You're breathing steadily. There's some fatty acids out there. So glucose is going to come in the cell, and we want to store the glucose. We want to store the glucose. So how are we going to do that? We're going to take some fatty acids. We're going to burn them. We're going to burn the fatty acids, the energy in a fatty acid, because the fat is actually a really excellent source of energy. It's a more energy-dense source of power than any of the other biomolecules. We're going to burn some fat, and we're using that energy to convert some ADP to ATP, using that ATP to catalyze the storage of glycogen in that long animal starch molecule that I talked about earlier. Meanwhile, we can also put some more phosphate in the bank with some creatine. We're just packing energy away from when we need it in the form of glycogen and creatine phosphate. All right? The output of this is all aerobic. This is the oxygen coming in, carbon dioxide coming out. Here's the carbon coming in. The under-oxidized, the highly reduced carbon coming in. So fats are fairly reduced. We're going to oxidize them. I'm not showing the water, but water is coming out of there too. All right. So now we're done chilling out, and it's time to dance. So let's start burning some glucose. We're going to mobilize the glycogen reserves. We're going to mobilize the glycogen reserves, and we're planning to mobilize our energy reserves in such a way that we're going to be burning the oxygen. Burning oxygen to do it. So get to the glucose, because this is energetically favored. We liberate the glucose, because we had to put energy in to form that bond, so we just cleave that, get the glucose, and then we start breaking this down we're starting breaking carbon-carbon bonds. We break carbon-carbon bonds. That's going to give us, so glucose is 6 carbons, pyruvate is 3 carbons, so we get 2 3-carbon fragments here, pyruvate and DHAP, but essentially 2 pyruvates, and that gives us 2 ATP. We didn't have to use any oxygen yet, no oxygen. That's called glycolysis. This is called glycolysis for those of you, probably at least half, I imagine, that have had that before. That's glycolysis. A lot of steps in there. Don't sweat them. Pyruvate goes into the mitochondria, our little bacterial buddies from a couple billion years ago. And now, this pyruvate is going to get broken down to carbon dioxide with the help of some oxygen. Much more efficient. Boy, these bacteria know what they're up to. That gives us 34 ATP. Way more efficient, way more energy efficient, right, than glycolysis, only giving us 2 ATP. The whole process is going to yield 36 ATP per glucose molecule. All right, so now this is not like a casual jog in the woods. This is not a little bit of fun ski skating or whatever. This is a werewolf senator from hell is coming to tear your heart from your breast, and you are ready to get the hell out of there. Anaerobic metabolism, peak activity. So the bacteria, the mitochondria, I shouldn't call them bacteria, they're not, they're organelles. They can't keep up. Actually, we cannot get oxygen to the cells fast enough at a certain point. Our cardiovascular system just can't transport enough. We exceed what's called our VO2 max. The volume of oxygen our cardiovascular system can transport. And so we're left with the anaerobic portion of glucose hydrolysis. For every glucose, we're just getting 2 ATP out. Build up a lot of pyruvate, which becomes lactate. Lactate gets exported to the blood, recycled in the Cori cycle at the liver. And now, this werewolf senator from hell seems like a good time to take the creatine phosphate out of the bank. So we start burning phosphate that we had stuck away for a rainy day. All right. Anaerobic metabolism. So here is a, do I have a, oops, no. Here is a summary of all that. Here are a couple different phosphogenic elements. So I have ATP. There's just ambient ATP in the cell to begin with. So that's the first thing we burn. And then there's creatine phosphate, the phosphogenic system. That's a short term. This will give us 10 twitches. We have 10 twitches of ATP stored in a muscle cell. We've got 70 twitches based just upon creatine phosphate. This will get you 2 seconds down the road. This will get you about 15 seconds out of town. Glycogen, anaerobically. If you're just going to burn glycogen like there's no tomorrow, you can get about 2 minutes out of it. About 2 minutes. And so this is why weight trainers, there's a couple of you, that must be, some of you in this class are weight trainers. How long do you do a rep during weight training? So a set of reps, what is that? That's like maybe 10, 15 of them, takes a couple minutes, right? I don't know. Yeah? This is why. This is why. Because that's how long it takes for you to burn the glycogen in that cell. Anaerobic. You're trying to increase anaerobic capacity. So what you want to do is burn out the glycogen in that cell to cause that cell to adapt to be able to make this longer. That's what weight training is trying to do. Next, aerobic oxidative. I'm skipping the myokinase thing. It's not on this table, but it's part of it. It's more emergencies. Aerobic. So you can go about 40 minutes on the same gas in a cell if you burn it using oxygen. If you burn it within the confines of your VO2 max, that is your body's ability to bring oxygen to the cells, where the mitochondria do their deal. All right? 40 minutes versus 2 minutes. All right. So this is the same thing. It's just a different for people who like pictures of stick characters running around. I'm not gonna. This is the exact same thing. I want to talk about this oxygen debt. So, EPOC, excess post-exercise oxygen consumption. So you start out, you're running a sprint here. You're going to do the 100 meter or whatever. And you start out, first thing, you don't kind of ramp up to speed. You come out hell's bells. And your body is burning energy as fast as it can. But the VO2 work that your body can do, it takes a minute to get up to steady state. It takes a little bit to get up to steady state. Such that when you stop, there's an oxygen debt that you have to pay. That oxygen debt is helping to replenish the phosphagen system. It's helping to restore the other the whole phosphagen system, all the phosphogenic sources, to rebuild up the ambient ATP, maybe to make some more CMP or the CP. Yeah, the fact that we don't start breathing heavily to begin with is paid for at the end by heavy breathing at the end. All right? I don't know. I've never thought about it like this, but maybe if you right before heavy exercise, if you do really deep breathing and do oxygen saturation, increase oxygen saturation in your blood, maybe you can reduce oxygen debt and slide this curve to the left. I have to think about that. All right, oxygen debt. The Corey cycle, yeah. All right, so you know what? We only have a couple minutes here. Why don't you guys ask me some questions? Does anybody, what concept did I cover today that you had the hardest time with? Just tell me. Anyone want to volunteer a concept that they had the hardest time with today? Yeah. Sure. F-actin, F stands for filamentous, G stands for globular. Globular actin are just the beads, the individual beads, and F-actin is the chain of beads. The active site is on G-actin, and there's a series of active sites on F-actin. They are all attached to nebulin, which is the core protein that runs down the middle, and that active site on the G-actin is covered by troponin under the regulation. No, it's covered by tropomyosin under the regulation of troponin. Did I say all that too fast? Tropomyosin is a protein whose whole job it is to just cover the active site and prevent myosin from binding the active site. And it does that under the regulatory control of troponin, which is sensitive to calcium. Calcium's the real trigger. Yeah.
So is that what the act and then the slides?
Yep. Totally. Yeah. Actin, it was like Alan, and I don't remember the other actin molecule out there, but they weren't moving. Yeah, Casey, they weren't moving. They weren't doing anything. It was everybody else that was doing the motor protein thing along the track. Yeah, actin is the track. Yeah. Nebulin is the attachment protein down the middle. Tropomyosin is not really holding anything together. It's covering the active site. Yeah, it's a protein that covers the active site. Anyone else? Okay. Well, that's it.